Hello everyone. What do you feel when you see this kind of green color picture? No doubts, you all will love to visit this kind of place. Under this green cover, there is a material called soil like this, and which is dark in color and high in nutrients that to support those green plants. But over time, some of above green lands became agricultural lands like this, which is not much bad. People cultivated in lands with the purpose of finding food. Moreover, because of continuous agricultural practices with improper land management, those agricultural lands became lands like these, which are degraded land. But if these actions continue in near future, these degraded lands will be like this, which is extremely bad for existence of animals and humans. Therefore, under this situation, I would like to recommend green manuring as a better solution for under a sustainable agriculture concept. This picture shows how tropical countries do their green manuring. This picture shows how temperate countries do their green manuring. There are many green manure crops, but among them, I would like to recommend Sunnhemp as a good green manure crop. But thing is, the best soil incorporation stage of Sunnhemp under Sri Lankan condition is not well explained in scientific literature. Therefore, my study was to identify the best soil incorporation stage of Sunnhemp as green manure. Good morning, everyone. Let me introduce myself. I'm S.D.C. Maduwanthi from Department of Crop Science. What is green manure? Green manures are plant materials incorporated into soil while still green, basically to improve soil fertility and land productivity. The main crop under my study was Sunnhemp. Sunnhemp belongs to family Fabaceae. It is a legume plant with a short life cycle. It grows in warm season and it is a short day plant. Why Sunnhemp is considered as a good green manure? Because it is well adapted to a wide range of soils and well known to suppress nematodes, improve soil properties, reduce soil erosion. A fairly rapid growing plant, it takes a very short time to decompose. General objective: to identify most effective stage of soil incorporation of Sunnhemp as a green manure under Sri Lankan condition. Specific objectives: to determine the different levels of biomass addition to soil by Sunnhemp at different stages, to estimate the different levels of nutrients provided from Sunnhemp plant matter at different stages, to estimate the different levels of nutrient addition to soil by Sunnhemp at different stages, to identify the best stage of Sunnhemp soil incorporation as a green manure. Let's move on to the materials and method. I selected Mahilupalama Subcampus, Faculty of Agriculture, University of Peradeniya as my experimental location, and I conducted it as an open field experiment. As the very first step, I did land preparation. Primary, secondary, and tertiary tillage of the land was done using disc plow, a harrower, and rotary tiller. This was my field layout. There were 4 blocks, 4 treatment stages, altogether 16 plots. There was a maize border all around my field. After that, I broadcasted Sunnhemp seeds with the rate of 100 kilograms per hectare. Irrigation was provided by sprinklers. Manual and mechanical weeding was done by me. Under this presentation, this is a key point I would like to highlight. These are the 4 treatment stages under my study. The first one was T1 treatment stage. It was emergence of primary inflorescence. It was observed 40 days after planting. T2 treatment stage was emergence, primary inflorescence completely emerged, but no bloom flowers. It was 47 days after planting. T3 treatment stage was minimum 1 bloom flower is present in the primary inflorescence. It was observed 50 days after planting. T4 stage was completion of flowering in primary inflorescence. It means 63 days after planting. At the relevant harvesting stage, the plants were pulled out with the roots and chopped by a knife like this. Those pieces were distributed all over the plot. After that, mixed with soil. This is a picture of the field after harvesting several treatment stage, and biomass was allowed to be decomposed at each each and every different plots. Under data collection, I would like to go with soil sample data collection and biomass sample data collection. To select biomass samples, I used 50 by 50 centimeter quadrant frame, and plants were carefully pulled out. Then I measured fresh weights, dry weights of those biomass samples. After that, I did plant nutrient analysis. For soil sampling, I collected soil samples at harvesting, 2 weeks after harvesting, 4 weeks after harvesting at 0 to 15 centimeter and 15 to 30 centimeter soil depths. Then I did nutrient analysis for soil samples. In the laboratory, total nitrogen, potassium, phosphorus, organic matter, calcium, pH, EC were measured in the soil samples. In plant biomass samples, nitrogen, phosphorus, potassium, and calcium were measured. Data analysis. Collected data were analyzed with the analysis of variance procedure using the SAS statistical software. The mean separation was done using Duncan's multiple range test to identify differences between treatments. Let's move to the results and discussion. First of all, let's look into the variation of plant dry biomass with time and at each treatment stage. With the treatment stage, plant dry biomass was increased gradually. It is same in the weekly basis also. Gradual with time, plant dry biomass was increased. The same pattern was given to the plant dry biomass nutrient content in nutrient analysis. Nitrogen, phosphorus, and potassium and calcium. Roughly, all 4 nutrients gave the same pattern of increasement with the relevant treatment stage. Variance of organic matter at 0 to 15 and 15 to 30 centimeter soil depths with time. Now we have moved into the soil sample results. So this is how 0 to 15 centimeter soil depth, and this is how the organic matter content has varied at 15 to 30 centimeter soil depth. In both soil depth, with the time, organic matter content has been reduced. It has been simply elaborated in these 4 graphs separately, one treatment to one graph. As you can see here, in 4 graphs, it means in 4 treatment stages, organic matter content was reduced with time. This is how 15 to 30 centimeter soil depth, it means lower soil depths varied their organic matter content at different treatment stages. Variation of soil nitrogen at different sampling stages. Even after 1 month of time period, the nitrogen content in soil at 0 to 15 and 15 to 30 centimeters soil depths were higher than the initial nitrogen content in both soil depths. We can simply elaborate that paragraph with these 4 graphs. Variation of nitrogen with time in 4 treatment stages at 0 to 15 centimeters soil depth. In treatment 2, 2 weeks after harvesting gave the highest, and treatment 3, 2 weeks after harvesting gave the highest. In treatment 4, at harvesting stage, sampling stage, gave the highest nitrogen content. This is how 15 to 30 centimeters soil depth vary their nitrogen content at, to 4 different treatment stages. These 2 graphs shows variation of soil phosphorus at different sampling stages. And this is how soil potassium vary at different sampling stages in 2 different soil depths. Conclusion. T4 produced significantly greater plant dry biomass compared to all other treatments. And however, the time taken for T3 was 50 days after planting, and T4 was 63 days after planting. In terms of nitrogen and phosphorus levels in plant dry matter, both T3 and T4 showed significantly greater levels compared to other two treatments. The potassium and calcium levels in plant dry matter was significantly greater in T3. Percentage organic matter addition to soil at 0 to 15 and 50 to 30 centimeters depth was significantly greater during harvesting and 2 weeks after harvesting in T3 compared to T4. The soil nitrogen levels at 2 weeks after harvesting was significantly greater in T3 compared to T4. The soil nitrogen levels at T4 at 4 weeks after harvesting was significantly lower both in T3 and T4 compared to 2 weeks after harvesting. Let me quickly go through conclusion as a summary. T4 adds more biomass to the farmland but takes 63 days for soil incorporation. But both T3 and T4 add similar levels of nitrogen and phosphorus to the soil. Nitrogen addition to soil by Sunnhemp biomass reduces with time. Thus, according to overall results, in terms of nitrogen addition, T3 would be better. In terms of plant biomass addition, T4 would be better. Due to the longer time taken for T4, farmers can incorporate Sunhemp to soil between T3 and T4 treatment stages. The cash crop planting between harvesting to T3 to 3 weeks after harvesting of Sunhemp will provide more soil nitrogen to the growing crop. These are the references I got information. And thank you for listening to my presentation.